Thank you all for joining today. We're excited to have you all back. This is our follow-up question and answer session to our November 1st webinar on land-applied biosolids and PFAS exposure. You may recognize me from that event. I'm Rebecca Eicher, a project director with the AAAS Epicenter. The Epicenter works to make it easier for decision makers to access and use scientific information and for scientists to engage with policymakers at scale. We work to bring clear, concise, and actionable scientific evidence to decision makers and build trusted relationships between scientists and policymakers. So thank you all for joining us, and thank you to our panelists for coming back. During our virtual event today, we will be talking about per and polyfluoroalkyl substances, or PFAS, and land-applied biosolids. Because of the strong interest and the high volume of questions asked during our previous webinar, Land-Applied Biosolids and PFAS Exposure, we're hosting this follow-up to address questions that came in during that webinar. This will be an opportunity to delve more into those specific issues, address some of the technical questions that were posed, and for the audience to ask clarifying follow-up questions during the panel discussion. There were many questions posed during the previous webinar, and we have combined and reworded some of those questions to ensure we can get to as many questions as possible. Again, we cannot promise that we will address your specific question, but we hope this discussion provides additional clarity and answers many of the questions that we did receive. Also, some of the questions that were posed during the webinar do not fit within the expertise of our panelists today, so we won't be able to answer every question posed previously. We will be able to address new clarifying questions, but probably we won't have time to get into any new topic area questions around this. So we will be really focused on what already came in during the previous webinar. We are thrilled to have Drs. Andy Smith and Tom Simmons from Maine CDC, as well as Dr. Mohammed Atia Ibrahim from the Office of Research and Development at the U.S. Environmental Protection Agency. Dr. Ian Pepper is not joining us for today's follow-up session, so we won't be able to address any of those questions that came in specific to his studies. I'm excited to get started with the discussion, and just to kind of orient you to the order of questions, we're going to start with those questions that came in specifically about Maine CDC's study and paper. Then we'll move to those questions about analytical methods, and then we'll get to some of the more general questions about biosolids, modeling, guidelines, exposure questions. So that's our plan. I do want to ask in advance for your participation in our survey at the end of today's discussion. It should only take 2 to 4 minutes to complete. It's anonymous, and your responses are incredibly helpful to know if we're meeting your information needs. The survey link will be put in the chat towards the end of today's discussion, and it will appear when the webinar ends. So thank you in advance for filling it out. So let's get started. Welcome, Andy, Tom, and Moha. I will start by saying, is there anything you guys wanted to say to kind of kick off the discussion, or should we just jump right into questions?
Jump right in.
Yep.
Okay, I will also apologize that I may have to mute to cough. I have a cold, and so I don't want to cough in anyone's ear while we're doing this. So as we jump right in, we know from the work that Andy and Tom did in Maine, you did some work on soil screening levels. So I was wondering if you can tell us more about those soil screening levels and walk us through that calculation. There were some questions about what factors were taken into account, what PFAS concentrations were in groundwater, how is soil depth factored in. So if you could kind of clarify and help us walk through that, that would be great.
Yeah, certainly. And I think the easiest thing to do would be to share our slides that Andy presented last time and go to slide or page 4. So this is the soil screening level equation that was presented for the soil to feed to milk pathway. And I can quickly walk through this and then try to address some of those questions about other factors, groundwater depth. So this equation is a repurposed equation from the preliminary remediation goals for radionuclides, just for, instead of being radionuclide specific, PFAS specific. So those terms in red, you see PFAS action level, transfer factor intake to milk, and then the plant transfer factor are all PFAS specific transfer or PFAS specific terms. Our study, plant study, we looked at gathering more data for the plant transfer factor. We have two studies in the literature that give us estimates for a PFAS transfer factor from intake to milk for the cow. And then we need an action level. Right now, there's no federal action level for PFAS in milk, so we've derived our own. So those are kind of the three big terms that go into it for PFAS. The rest are terms that get at consumption rates for cows. So those green terms, the I fodder, fraction land, those are the ingestion rates for feed for cows. And then the fraction of land that's contaminated. For our soil screening levels, we're assuming 100% of the land is contaminated, biosolids contaminated land, but you can adjust those if you're in a site-specific assessment. And that last set of terms there that are brown, the I soil, ingestion soil, are really in soil ingestion rates if cattle are out grazing, and then the fraction terms there, you can adjust those for the time spent grazing for that. So that's overall the equation we use to develop soil screening levels for this agronomic pathway. We also have a very similar equation, if not identical, for the beef pathway, just specific for muscle to beef intake for PFAS and some different intake terms. As far as accounting for binding or anything like that, with the plant transfer factor, we can talk more about that in some other questions, but we don't take that into account directly. It's just a simple ratio of what's concentration in the soil to what ends up in the plant and concentration wise for the plant transfer factor. So no taking into account binding or anything like that. We don't look at groundwater in this model. So groundwater is exposure pathway is separate. There are a couple other questions of getting that percent moisture. We can talk about it later, but we don't look directly at percent moisture in this model either, if that has any impact with soil or the percent moisture of feed intake. And then one of the biggest ones for the plant transfer factor is soil depth. So all of our studies are looking at a 0 to 6 to 0 to 8 inch roughly depth. And that's not to get at what the roots will be mostly in contact with. Some roots might be deeper, shallower. It's really for our studies to be applicable to what's happening in the state for other soil testing. So we have our colleagues at the DEP out testing 500 to 1,000 fields at about 0 to 6, 0 to 6, 0 to 8 inch depth. So we have all of this data coming at us at that depth, and we need to be able to translate our studies findings for plant transfer factors to other fields. So that's really where the depth comes into play. It's not so much about understanding the roots structure and that depth. And I think that's covers most of it. If Andy has anything more.
No, I think that pretty much covers everything with that model. Other than, I don't know if you're going to mention, Tom, that we've been surprised that if you're in a situation where, you know, all the feed is locally sourced on the farm and all the fields are impacted, where you're able to model a period of time where the animals are just consuming that, this model seems to actually do quite well. It's easily within a factor of 2 of observed milk levels and what we would predict by the model. So it gives us some comfort for a screening level where rarely our soil screening levels, think of like the residential incidental soil exposure pathway, model checked as much as we've done model checking with these.
Great. And then I think one of the additional questions came in really specific about Maine and whether it has any karst areas. And if so, what fate and transport occurrences have been observed in more basic soils and carbonate aquifers.
Yeah, I can't speak specifically to karst areas in Maine, certainly not my expertise for the geology in Maine and where we've conducted these studies. We did look at soil pH as part of the soil fertility parameters in our studies, our plant uptake studies. And we don't see a correlation with pH and PFAS uptake. So pH ranged from about 5.6 to 7.8 in the soils. And we don't see any correlation there, whether increasing or decreasing for the uptake of PFAS into plants. So at least for pH, we don't think there's a correlation there with uptake.
And then Rebecca, if we could advance to slide 7, I just want to make a comment about that slide as it relates to the soil screening level. So they may remember this is the range we gave, and I just want to emphasize again, that range largely reflects what you use for a toxicity value. So, and I think that's an important point out right now. There's, the sort of toxicity values currently in use are either the US ATSCRs, which for PFAS is 2 nanogram per kilogram per day. But then you have the new EPA updated tox value used for the basis of the National Drinking Water Standard. That's 0.1 nanogram per kilogram per day. So you remember that in that numerator of that equation is the milk action level, and that milk action level is going to be somewhat predicated on what you use for toxicity level. So some of this range you're seeing within a given type of forage is just differences based on what toxicity value you use. After that, it's largely based like corn silage versus corn snaplage on differences in the plant uptake term that goes in there and differences in the intake of that type of forage crop for beef cattle.
Great. And I think that was one of the follow-up questions that had come in. So thank you for preempting that. And then I want to ask this, recognizing that you may not have the expertise to answer this, but biosolids application methods became a question, and whether that could impact fate and transport of PFAS in the soils. So the example was, biosolids could be surface applied or incorporated into the soil. And how would one think about that, or how did you all think about that? And if you didn't, let us know.
Yeah, for that, we did not think about how the biosolids were applied. Our understanding for most of the fields in Maine, it was surface application, and this will all be historical surface application. So in the decades that we've had a biosolid spreading program, when we come to farms, it's now all been applied. There's no current ongoing application. So it's really about what's existing now in the soil from historical application. So we didn't look at any application rates or differences in types of application in our studies, just what's in the soil.
And another complicating factor is we're talking about land application of biosolids over decades. So the actual use of the fields may have changed. So whereas there may have been a period of time when it was applied to the surface for a perennial grass-based forage crop, that field may have formerly or more recently been converted to growing, say, corn for silage. So now it's applied and tilled into the soil each year for depth. So it can really vary from field to field depending on the historic use of that land. And we've definitely encountered that in the field where we're on what used to be cornfields, now it's grass-based, or the other way around.
Yep, got it. Moha, I don't know if EPA has done any looking at this or anything you would want to share about that.
So I would just echo the exact same message about the unknowns, but we actually do have a colleague here who is looking into that on a field scale. I also want to confirm about the issue with the historical use of the fields themselves and different crops, etc. So there are so many moving parts, but there has been data being collected over the past 3-4 years, not yet published, but I think when that gets cleared, I guess this will answer or try to fill some of these gaps.
Great. So the ongoing work. The next question that I think might be on some folks' minds is whether there's a way that we can try to understand what levels in soil mean for levels in groundwater. So is there a way that folks could kind of think about this in a general multiplier example or situation? For people to understand this is a high level in your soil, very contaminated versus maybe less contaminated. I don't know, Moha or Tom and Andy, if you want to tackle that.
So it's very general. So if there is if the question is seeking a general answer, I would say no. We cannot really get that one answer that can just satisfy all the missing points. On the other hand, I guess it is. Sorry, that. So that on the other hand, it is. There has been a lot of work when it comes to modeling. So if you do proper site characterization, knowing what is the soil type, knowing what is the organic content, knowing like precipitation level, groundwater level, so that can be fed into a model that can help you predict it. But without that, I guess it will be very hard.
And also, if we could bring up slide 12, I think the point I would add is it's not an area that we are working in at Maine CDC, but our DEP colleagues are working with other states and some academic centers to sort of look into this whole issue of trying to model the movement from soil to groundwater. And we have to appreciate that modeling and even thinking about that's going to be very different depending on where you are. So for example, here in the Northeast, probably about 3/4 of our wells are going to be bedrock wells with, so fractured bedrock wells with not a lot of cover, overburdened cover between the soil surface and the groundwater or the bedrock. And that's going to be very different in terms of thinking about how much is too much in soil for a groundwater pathway than say someone who's got a much deeper layer of soil overburden and may have sand and gravel aquifers as opposed to fractured bedrock. And just to sort of illustrate that, I showed this figure when I gave my talk of looking at wells that have been tested in close proximity to farm fields with known to have elevated levels and showing that you can get some really very, very high levels here in Maine in groundwater. But also note how some wells right adjacent to fields have very little. And that just has to do with the actual directionality of the fractures that are in the bedrock itself. So you can imagine this is just not going to be an easy situation to come up with a one number easy conversion.
Yeah, great. Well, thank you. And Tom, I think you kind of referenced the historical context of some of these fields, but we did have a question whether your studies or any that you know of evaluated how PFAS can build up or accumulate in the soils over time with continuous application. And would the soil screening levels be variable on a field with continuous application? How would crop uptake impact that? Any sort of thoughts on that you could elaborate on?
Yeah, we haven't looked at that specifically in our study. Again, we're coming to sites that are already contaminated from that historical application. So we're not looking at ongoing application and additional contamination, accumulation, or depuration out of those soils from that. And I think at a higher level, what we can say is, at least in Maine, there are some fields that have a longer history of spreading that can sometimes have higher levels. So if you're just thinking about application rates, and this goes back from decades of spreading, so it's hard to know exactly what was in those biosolids at the time, but a longer history of spreading can sometimes have higher concentrations in the soil. And certainly we're seeing when we go back multiple years to sample a plot, for example, we don't see much depuration or change in soil levels for a lot of the PFAS we're measuring. So at least over the short time frame that we've been out multiple years now, there doesn't seem to be a large depuration from the soils once you have that contamination.
And if this question is also trying to get at, is there a difference, for example, in plant uptake of PFAS contaminated biosolids being recently applied and still being incorporated into that soil matrix versus long ago applied and therefore much more sort of integrated, homogenized, whatever, with that soil matrix? That's an interesting question, not one that we've looked at and have any information on, but it's an interesting question. So.
Great, thank you. Okay, so Moha, anything from EPA on that front, or is that not something that people have been working on over there? Oh, you're muted.
You'd be surprised how much work done on that internally, but it's still, I guess, most of it is not published yet, so there's no conclusive answer about it. But the good news that it's in the pipeline.
Great, thank you. Okay, well, I think that's really helpful and addresses many of those soil screening, soil contamination questions. So I was thinking now we could move to the soil to crop uptake, cattle, milk, human work that you all had been doing. And maybe it would be helpful to just quickly remind people of the study that you all did in Maine and the main take-home messages from that. And then we can address some very specific questions that came in about that portion of your work.
Yeah, certainly. I think maybe if we go to slide 3, just to kind of set up the overall pathway again, and then some of the plant uptake work. And so if you remember back to Andy's talk, a part of his first section of the talk was looking at this pathway from soil to feed to cow and then the milk and then to the consumer. And one thing that to model this pathway that we need is that uptake from soil into feed. And at the time, at the literature, there wasn't much for biosolid application soils that were contaminated and uptake into plants. So our study focused on getting more information specifically for that plant transfer factor on fields that we knew had a history of biosolid application. So if you go to slide 5 now. So this was just our general study design. This looking at fields with varying soil concentrations for PFAS here at one farm. We did a small research plot study with 4 plots across the fields for our grass uptake. We also looked at several other farms in Maine to get a better sense of different biosolid histories, different soil contamination concentrations for PFAS and uptake into grasses. So we do have some of that work as well. But basically go out in the field, collect a co-located soil and grass or soil and corn plant sample. So it's basically a soil sample right below what we're going to harvest for our grass in a 1x1. And we take all that grass that gets shipped off to the lab. We go into the 1x1, take a core, a 6 to 8 inch core for a soil that gets homogenized, gets shipped off to the lab. For corn plants, if you can see there, we actually take individual corn stalks. For corn silage, we take the whole stalk above the ground, cut that up, and that goes off to the lab for analysis. For snaplage, which is just the husk snapped off of the stem there, you get the cob, kernels, husk, silk, and some of that shank. And then we have looked at earlage with just the cob and kernel as well. And we wanted to better understand what the transfer factor would be for these forage crops to use in our model. So if you go to the next slide, slide 6. Can you put the chat on at the side? Sure. So this is looking at the results for the PFAS transfer factor for grass-based forages, the green box plot there, and then some statistics below for the field average transfer factor and then with corn silage and snaplage. And one thing with grasses we noticed is you can see there can be quite a bit of variability for PFAS uptake across farms and fields. Ranging from 0.03 to almost 0.4 for a transfer factor. So quite a bit of range there for the PFAS transfer factor. Less so for silage as far as a range goes and snaplage, but it's interesting. We see less uptake into corn silage than we do for grasses, and then even less uptake into snaplage. So this gives us some information both to use in our model as well as some information to use as a tool for crop decisions for helping to reduce PFAS exposure at farms if they can switch from a grass-based to a corn-based operation for that. So that's kind of the overview of the study there.
Do you want to talk about first cut, second cut?
Um,
The next slide? Or did we even?
I don't think I put that in here.
Ah.
No.
What Tom and I were just chatting about is, if people have read the paper, they will see that there's also a figure talking about the differences between a first cut crop and a second cut crop. And we see considerably higher uptake into the second cutting than into the first cutting for some preliminary results we report in that paper. Again, that's for the grass-based forage crops. And just another factor to have to try to take into account in the modeling.
Yeah, great. Thank you. And apologies if you addressed this question, but just to clarify, does the amount of adsorption to organic carbon increase the uptake of PFAS into crops? Is that something you all looked at or have any information on?
Yeah, certainly. So it is something we looked at in our grass uptake study as well as our corn study. But for the grass uptake study, we didn't see a significant trend of increasing organic carbon and decreasing PFAS transfer factor uptake. There was that trend, which we see in the literature as well for other studies with higher organic matter in soils, there can be less uptake. But for our study, it didn't reach statistical significance for a correlation between the two.
Great, thank you. And then I think,
Lots of interesting chat, which, in a bunch of these, we don't really have the answers to, but we really appreciate them. There's a lot of interesting questions and being asked in the chat, so.
yeah. And what I will say is that, well, I forgot to remind everyone that you should see that this is being recorded and we will make this available for everyone who attended, as well as it will be on the Epicenter's webpage. So you will have the chance to rewatch and go deep on this. And the questions that are coming in, again, we probably will not be able to get to any of these new questions, but we appreciate you asking them so that we can share them with Tom, Andy, and Moha, and then we can help think about some additional ways to address these questions, maybe additional webinars, opportunities to really share some of this. And speaking on behalf of Andy, Tom, and Moha, it does also help them think about some of the research questions that they could try and address as folks ask these kinds of questions. So we do really appreciate them, even if we can't answer them or get to them today. So thank you for being such a ambitious and motivated group. I think some of the other questions that had come in around this was then about kind of what you see when you go from the plants into the cows and into the milk. And so I was wondering if we wanted to jump to some of that work. And then, Moha, always feel free to share any perspective that you have from the research that EPA is doing and or supporting with the academic community. So Andy and Tom, is that okay to move to the milk stuff?
Certainly, yeah.
Okay. So I think one of the questions that came in is, would any PFAS precursors present in the feed and water contribute to PFAS present in milk? And if I can ask for you guys to pause and remind everyone, what are PFAS precursors? When someone refers to a PFAS precursor, what are they talking about? And then why would that be a question that folks are thinking about for it getting into the milk?
And Moha, do you want to take the first stab at that?
Can you repeat, because when you say transfer,
No, sorry. It's just it is about whether PFAS precursors can impact milk levels. And before we get to that question, I'd really like to make sure that everyone in the audience understands what PFAS precursors are. So what
Okay.
are the PFAS precursors?
Yes. So whenever we talk about PFAS, we just mostly focus on the terminal products. What does this mean? Like, there is a persistent form of PFAS that it is very hard to degrade. But when we talk about like the larger pool of PFAS, then they don't come from nowhere. They come from precursors. So precursors are parent compounds that has been used in different industries, and then based on their fate in the environment, if they go to soil or air or water, they might go through certain transformation that would lead to the formation to the final terminal products, or what we call them, terminal PFAS. So precursors, as simple as the definition goes, is that whatever PFAS has been used in original form in the industry that can lead down the line to forming terminal products of PFAS.
And just to make sure everyone is totally clear, can you give an example of a terminal PFAS?
Yeah, so the famous C8 or PFOA or PFOS, this has been phased out, but these are kind of famous because they're also among the regulated PFAS in drinking water now. So that's when we say like a terminal product, meaning that there is no living organism or anything that we know of yet that can actually deal with it in the environment, and that's why they kind of persist. And this is one of the reasons when people talk about like forever chemicals, that's one of the justification of using such term. Of course, people in treatability work would argue against that, especially with destruction technologies. But when we say forever, it's mainly about like environmental fate. So there would be different branched form of PFAS that under certain conditions or after applying certain chemical treatment, they would transform into the terminal PFOA or PFOS or other similar compounds.
Great, thank you. And then Tom and Andy, are you able to share whether any PFAS precursors present in the feed and water might contribute to PFAS present in milk?
Yeah, we can talk about that to the extent that we've looked at a specific set of precursors in soil and plant and milk for PFAS. So we have measured in our plant uptake studies in soil and plant PFOSA, N-ethylfosa, and methylfosa, which are three precursors to PFAS, PFOS. And what we see in our study, at least, is there can be concentrations in the soil for those precursors, but we don't see much of those precursors moving up into the grass plant itself. What we do find, though, is if you have high levels of these precursors, PFOSA and ethylfosa in particular, in the soil, we sometimes see high levels of PFAS transfer factors. So there's something going on there with whether it's biotransformation occurring at the root system, within the root, and leading to higher uptake of PFAS. And that could be impacting PFAS uptake. And then once it's in the plant, that can go into the milk eventually for PFAS. We haven't seen much for movement from soil at the plant to milk itself for those precursors. There is another set of precursors, FTSs, fluorotellular sulfonates, that we do sometimes see move from soil to plant to milk as well as beef, in particular 8,2-FTS. So it really depends on what precursor you're looking at and whether it will move through this pathway. And it looks like to us, very PFAS specific.
Great, thank you. And thanks for clarifying. Let's see.
While you're looking, Rebecca, I see a question about does Maine still allow biosolids containing PFAS to soils based on contamination from the past? At this time, Maine's legislature has banned all application of biosolids, period. So at this point, it's all going to landfills or other places, but there's no land application currently in Maine for municipal wastewater treatment plant sludge.
Thank you. Thanks for addressing that. I know that's a big question for folks. And different states have different regulations around this. So there isn't a national standard or rule for PFAS and biosolids land application as of yet. One question that has come in a couple different forms, but maybe Tom and Andy, you could just share. We know there are thousands of PFAS, and so you can't study them all. And so why did you start with the PFOS, PFOA? How did you hone in on which of these? And one set of compounds that a lot of folks have been talking about are these DIPAPs, which if any of you would like to tackle DIPAPs, we can put that as the second piece of that question. But Tom and Andy, if you want to just give a quick rationale for which compounds and why you studied those.
Yeah, I think for us, it's been what is the suite of chemicals that are being tested at the time with the current methods we have, and what we're seeing in the resulting agricultural food commodities that would be consumed. So, for example, when we look at milk, we mostly just see PFAS. Sometimes we see low levels of others. There's been a few isolated cases where we've seen some PFDA and PFNA in milk as well. Rarely do we see PFOA, PFOA. In beef tissue, again, mostly PFAS, sometimes PFDA, sometimes PFNA. And as Tom mentioned, we've seen these 8,2-FTS. So we're somewhat limited, sort of like the proverbial drunk looking under the lamppost for the keys. We're limited to the suite of chemicals that are currently in the test battery, and we've focused our work on those that we're seeing in milk or beef and at levels of potential concern.
And then I'll let Moha take the DIPAPs. We haven't done anything with that.
Yep.
So the first thing, Moha, is what are DIPAPs? And don't go deep yet. Tell us a little bit about what DIPAPs are and why we're hearing so much about them in wastewater situation.
They are weird looking PFAS where you have phosphonium in between. So I have two branches that can be cleaved under oxidative conditions. So there has been some studies a few years ago that found them in soil or biosolids. And then tracking that back, that just was linked directly to wastewater treatment plant and sludge in there. And then more specifically, I guess some people are linking them to consumer products. So in some of the studies that we run, we see it very clearly in domestic wastewater. One of the studies, famous, it made noise. I don't remember last year or the year before that it was found in toilet paper, and I guess that's one of the potential main sources in domestic wastewater. So in a study that we published recently, actually targeting landfill leachates, one of our control samples was for a domestic residential only kind of wastewater, and DIPAPs was there. And then after applying top assay, I guess some of the questions are about that. I can explain later, where you oxidize the sample to transform the precursors into terminal products. you can see an evidence of just like so many different precursors in there. So in a follow-up study that we are wrapping up now and hopefully should be published soon, that's only focusing on the septic samples and residential residential wastewater. That has, again, another line of evidence that DIPAPs are prevalent there in both septic and pump stations. So that's DIPAP. So they travel all the way from consumer products to domestic wastewater, going to the wastewater treatment plant, going to the sludge, and if you apply that, that's I would assume one of the pathways that they reach soils. Yeah, I saw the question about tracking them. I'm not aware of anyone tracking them into just like uptakes, etc. I just personal hypothesis. I don't know whether they would be actually among the favorite molecules to be get uptake by plant because they're kind of big, and then I would think that maybe plants would prefer shorter, more mobile kind of structures. So yeah.
Okay, great. And then I'm going to ask one more question for Andy and Tom, which came in around who is responsible for testing PFAS in milk? The state ag department, FDA, USDA? How do people think about who is measuring PFAS in milk? And are you all willing to answer that question?
Well, I will answer it with the caveat that it's sort of outside our wheelhouse, so I'll give you my best understanding. But when it comes to milk in the national food supply, that's FDA's jurisdiction. So any testing would be FDA. I believe they do testing as part of the total diet survey, and you can go online at the FDA website and actually see that data and what those levels they're finding as part of their testing of the national food supply. Separate from that, states who are implementing the food safety code have the ability, if they so choose, to test milk and take action on milk that is produced, processed, and sold within their state. It just, once it gets to be an interstate issue, that's FDA's domain. That's my understanding. So in Maine, we've done testing of retail milk. We've actually, our ag colleagues have done 3 rounds of testing for retail milk. So we have some sort of sense of what we'll see. Levels were, in general, mostly non-detect, with a few detects. That's actually how we found one of the farms, is you can actually trace back from a retail sample to what farms likely contributed to that sample. And again, that's how we found one of the farms. So yes, states can test if they so choose. And in Maine's case, we have done that.
Great, thank you. There were a lot of questions and shared information about some of the research that Andy and Tom have done related to PFAS in eggs. I'm gonna move on from that, even though it's very interesting, and I know we have lots of good stuff to share, but yet again, we don't have all the time that we wish we could. So I want to talk a little bit about current methods of detecting and quantifying PFAS in biosolids, as well as in influent and effluent at wastewater treatment plants. And so Moha, if you could help us think a little bit about what are the current methods, and then you had sort of referenced some of the leading or emerging methods on detecting PFAS. So could you tell us a little bit about influent, effluent, and biosolids PFAS detection tools?
Yeah, the shortest answer ever would be like EPA has finalized and published recently the method 1633. So that's definitely the again the direct answer for the 3 of them. But that comes also with some limitations that should be highlighted, that even though very sensitive method, very good for complex matrices, but at the same time, it only covers 40 PFAS. And then just a reminder that we talk here about like thousands of different structures. So the 40, even though they can be very informative to us, we cannot just like make a final understanding about just like the condition of PFAS in a certain sample. And that's why there are, as you call them, emerging, even though they have been around for a while, but the exact same method that I was going to explain, the total oxidizable precursor. There is no standard method for it yet, but this is one of the projects that we are actually working on, trying to get a standardized method for top assay, which is basically an indirect indication of the presence of or absence of precursors. So what we do, that you use a method to measure targeted PFAS, and then you apply an oxidation step so that can transform, as I explained earlier, the precursors into terminal products that you can measure. And then based on the difference like between before and after the oxidation step, you can get a hint about like, okay, there is much or less precursors in your sample. But at the same time, it doesn't tell you what are these precursors, but from an information or added extra element to our understanding about the sample, I guess is very informative. There are some methods in development for certain precursors that you can measure as well, but these would suffer from sensitivity levels. So like, even though you would have a method to target and measure certain compound, but then is it sensitive enough to go to the level that you can find in the environment? That's work in progress. So that's, I can talk about methods for much longer, but I guess for at least from my personal experience with this project that we have related to wastewater treatment plant, wastewater and associated samples that can be solid or liquid, I guess a combined extended list of targeted methods, if you combine that with top assay, you can get a fair good information about the sample and the PFAS levels that you'd have there.
Great. That was very helpful. And a question that had come in previously, and you can decline to answer this if you so desire, is whether method 1633 is underestimating PFAS amount.
Yeah, I think I just said that. I mean, like, it's an inherent problem. So you cannot say that the 40 compounds are everything that you can see. So there would be under certain condition, there will be an underestimation. And that's why I said, like, if it is combined with another method that can give us a broader view of potential presence of other compounds like the top assay, I guess that together you would be just filling different pieces of that big puzzle, basically.
Yeah, thank you. And then what have you seen in your research or in published research around the relationship between what people are sampling and finding in the influent, in the effluent, and then biosolids? Is it what comes in is what comes out, or is there differences in what types of PFAS are in the effluent, the biosolids? Anything you could share about that?
So there has been an observation that PFAS coming out is in the effluent is higher than the PFAS going in the influent. But now that people are extending the analyte list that they are targeting in terms of PFAS, and also using other methods that can get like an aggregated method like top assay, we know that that's a precursor issue. Like, you have stuff in there that have been overlooked, but now we can actually get a better understanding of what's actually happening. So they would partition some, like more hydrophobic stuff would partition into biosolid, and that's one of the potential routes of leaving the plant. But some of them would just, like, again, transform. If you have an oxidation step in your treatment train, that will transform them into terminal products. It can be microbial or other oxidative methods in the treatment train. So based on the efficiency of the treatment steps that you have there, you can get certain removal percentage, or you can get them out in the effluent. So that's the current. I guess we have a big project also being in the writing phase now that actually looked into multiple major treatment facilities around the states and going step by step within the treatment plant and consistently observing that reproducible observation about certain PFAS and transformation within the treatment plant are happening, and you get that partitioning in solids, etc.
Great, thank you. Okay, so transitioning a little, now that we all understand the methods for sampling, and we're all ready to go out and take our own PFAS samples, can you share a little bit about studies that have sampled surface water, groundwater, or drinking water near areas with land-applied biosolids? And if there are any generalities to the results from any of those studies. Tom and Andy, you might have something to share from how Maine approaches it. But again, it may not be any details of specific patterns, but just if you could share a little bit about how Maine thinks about surface water, groundwater, and drinking water near land-applied biosolids areas.
Well, I think that slide I showed earlier of, I think it was number 12, and just making the point that by statute, Maine is testing all sites in the state with a license history of land application. So that's where the soils data are coming from. But then they also have a mandate to be testing wells that are in adjacent to those sites. So we've got lots of data coming in, but I don't think anyone's really looked at it in a generalized way. On that state of Maine map you're seeing, those are all wells that have been tested that under this. So that means they're wells in close proximity to fields with a history of land application. And you can see there's clearly that cluster of high levels where there's known high soil levels. But if you're concerned, as we are, about wells in that 20 to 40 to 100 PBT range, we're finding plenty of those in fields that are in the wells that are adjacent to fields around the state. So I don't think we can say much more than that. And again, remember, this is through the lens of a state where most of the wells are drilled bedrock wells, as opposed to a place with a lot of sand and gravel and a much deeper depth from soil to bedrock.
Yeah.
Do you want to talk about surface water?
Oh, for surface water, we've certainly encountered surface waters that are, for example, ponds on farms that can have some fairly elevated levels, or streams that are running adjacent or through fields with contaminated soil from biosolids, where we'll see elevated levels in the surface waters. I think I had a slide on that in the talk as well. And that have resulted in elevated levels in fish and fish consumption advisories. So.
Yeah. Very
but
helpful.
again, nothing broad or generalized about that. So, and I don't think we understand how much of that is actually surface runoff versus subsurface groundwater moving into these streams.
Yeah. And maybe you can clarify for folks, because I think some people have, heard that in Maine, some of these biosolids had come from paper mills, paper facilities. But I believe in your previous talk, you had shared that not all of this was from what people might describe as industrial biosolids or industrial sludge, that there is some data or pattern to biosolids in general, not just those that have industrial.
Yeah, I think if you go to slide 8, and you'll see the results thus far from the testing of sites for the history of land application of biosolids. In the map, you're seeing the location of those sites in a range of concentrations. And, that red cluster is indeed a municipal wastewater treatment plant, but one that had significant influence from a paper making facility that made food molded type paper products. And that's probably what's going on there. But that, we think, is more the exception than the rule for Maine. think about Maine as having, as I understand it, three sort of broad categories of biosolids that were applied. I just gave you that example, which I think is more the exception than the rule. There was a fairly active program in the 80s and 90s for paper mill sludge and bioash from that was applied around the state. But there also was a very robust program for land spreading of municipal, typical sort of municipal wastewater treatment plant sludge. During that period of time, there was very little sludge going to landfills. So when you look at that map, you need to be thinking that it's representative of that whole mix. And think about also the potential for any of these to give you soils in that 2 to 20 ppb range, because that's the level where if that is being used to grow forage crops for animals that are going to produce food commodities that are going to be sold locally, you're going to want to be testing and you're going to be concerned with that. And maybe if we could just quickly go to slide 23, because I think this gets to the question as well. Nope, is there no, yep, 23.
Oh, there's only 21 pages.
Oh, okay. Oh, sorry, it wasn't included. It was a backup slide. Basically, it was a slide that would show a farm that we've been doing work on. It's a small dairy farm, and it has soil levels in some of the fields that are, circa 1 ppb or less, kind of background levels. There's 2 fields in the 12 ppb range, and the dairy cows spend their mornings grazing on the low fields. They're milked. May go back on those, and then in the evening after milking, they'll go out on those 12 ppb fields. Those fields are only sourced, as we understand it, with municipal wastewater treatment plant sludge. And it's resulting in milk levels in the 20 to 30 part per trillion range, which, if you look at some of the new tox values, would be levels of potential concern. So that's an example of a situation where it's not industrial sludge. It's not even a biosolid that has a significant industrial input, as we understand it. It's getting these ranges in this sub 20 ppb range, and that seems to be enough to get you an elevated milk if we care about milk in that 20 to 40 part per trillion range.
Yep, super helpful. Okay, well, we've got a minute left. And obviously more questions came in as we knew that they would, and we of course tried our best to get to as many as we could. I think one of the general questions that now it's 3, but I'll just take an extra minute to hear from each of you is how you would help people think about biosolids as a significant source of PFAS exposure. We know that there are many sources of PFAS and that exposure pathways. So I think a lot of folks are wondering what should we make of PFAS in biosolids and human exposure?
That's a broad question. I'll tell you as a public health person, this is the way I look at it. First, people should be aware that for these legacy chemicals that we're most concerned with, the PFAS and PFOA so far in these, if you look at blood serum levels in the US population, they're decreasing. So if you go back around 2000, the average person in the US had maybe 30 part per billion of PFAS in their blood serum. It's now down to under 4. That's all occurring during that time and probably has something to do with getting PFAS out of various sort of food packaging and things like that. But it's been trending down. That's one thing to put into perspective. The other thing to put in perspective is if you look at FDA's testing of total diet survey, they're not finding much for it. So for the average person buying the food from a supermarket, the biosolids issue is probably not much of a concern to them. But if you're looking at a farm that has PFAS contaminated land, and especially if that farm is selling direct to consumers with this sort of locally sourced food, whether it's milk or beef, then those are the ones that we are most concerned with and we've spent most of our time thinking about. That's not to make it any less important, just that it may be a more localized issue than a broad national public health issue.
Very helpful. Moha, anything you want to add, or Tom?
Tom,
Only thing I'll add to Andy's great answer there is, at least in Maine, one of the exposures that we're finding that's probably the most widespread is groundwater impacts. So it's in those localized settings where you have that food exposure, the agronomic pathways, you do see at a larger scale some contamination in groundwater, and private wells in Maine are an important source for drinking water. And those can be highly impacted. So that's just another thing to keep in mind in those areas.
Yeah, excellent point.
yeah, it's for me just like I don't think we can dare to generalize that. But the good news, that's my positive part of my brain. Luckily, it's on now. That our analytical capabilities are getting very, very sensitive. So measurement, source tracking, and all of that can be achievable now compared to just like decades ago. So that can help making an informed decision about like go, no go when it comes to like waste coming to the facility or going out from a facility. So that I would think about it just like a case by case scenario. Like are all biosolids safe? I don't think that's the case, but are all of them problematic? This is also not the case. So like that generalization, I guess it's not helping in that context. But at the same time, just like when you give the very good example when people just like got worried in Maine about all the biosolids, while the case that made the news was just like very, very specific. That's one of the clear examples about like why we need to ask about data, numbers, and source tracking, so that we can make a better decision.
Great. Well, we really appreciate you taking the time to come back right before the holidays. I know it's a busy time for everyone, so we really appreciate all of you who joined for us, and Tom, Andy, and Moha for coming back and answering so many of these questions. And we will make this recording available, and we hope that everyone who attended will continue to explore and get some of their questions answered. And please do take our survey to let us know if this helped meet answering some of your questions. So really appreciate you all joining. Thank you.